Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by an independent educational grant from Otsuka America Pharmaceutical, Inc.
Hello, I'm Dr. Jonathan Barrett, Professor of Renal Medicine at the University of Leicester in Leicester, United Kingdom. Welcome to this activity titled Visual Insights, exploring the disease-modifying effects of anti-APRIL treatments for IgA nephropathy. Today I'm joined by esteemed colleague Dr. Sunil Udani, consulting physician at Nephrology Associates of North Illinois and Indiana in Hinsdale, Illinois. Welcome, Sunil.
Thank you, John.
Over the next 15 minutes, we're going to discuss the background of IgA nephropathy, the pathogenesis of IgA nephropathy, focusing on the B-cell pathway, and the role of APRIL in the B-cell pathway. I'll finish by talking about emerging agents targeting APRIL for the treatment of IgA nephropathy. Now I'm going to turn things over to Sunil to bring us up to speed on IgA nephropathy basics. Sunil.
Thanks so much, John. So, IgA nephropathy is a rare kidney disease. In the U.S., the prevalence of IgA is somewhere in the realm of 2.5 per 100,000 adults, but it is more prevalent in patients of East Asian descent. Despite its rarity, it is the most common primary glomerular nephropathy worldwide. Some of the difficulty is the clinical presentation can be variable, and ranging from gross hematuria or asymptomatic microscopic hematuria, specifically episodes of synpharyngitic hematuria, where there's gross hematuria with an upper respiratory tract infection. There's also varied degrees of proteinuria. Ultimately, we can only make the diagnosis with a kidney biopsy, and that information ultimately leads us to the state of IgA nephropathy and can give us some idea of activity, prognosis, etc. One thing that's important to note is that most patients in the U.S. have chronic kidney disease at the time of their IgA nephropathy diagnosis, highlighting the nature of this disease to cause injury without overt clinical findings. We have framed the pathogenesis of IgA nephropathy into what has been known now as the forehead hypothesis. This starts with the increased circulating levels of galactose-deficient IgA1 or Gd-IgA1. There's definitely a genetic predisposition, and ultimately this starts with mistrafficking of B cells from mucosal to systemic sites. The second step is production of anti-IgA1 antibodies. Either those are IgA or IgG. And again, there is a genetic predisposition. There's a component of molecular mimicry. And we think this is stimulated by viral infections, environmental exposures, food antigens, a variety of underlying causes. Once those antibodies are formed, now we have immune complex formation, and those immune complexes are circulating. And how it relates to the kidney is those immune complexes deposit in the kidney, primarily in the mesangium, that causes local immune activation and injury. That injury is mediated by complement activation, other cytokine chemokine release that ultimately leads to mesangial sclerosis, interstitial fibrosis, glomerular sclerosis that leads to the clinical manifestations of hematuria, proteinuria, and ultimately kidney function decline. What's missing from the forehead hypothesis is the source of galactose-deficient IgA to begin with. And this slide digs into that a little bit more. Just as other forms of IgA, galactose-deficient IgA is presumed to source from mucosal immunity. Ultimately, what happens is that antigens are presented to antigen-presenting cells in the mucosa, such as dendritic cells. And specific antigens ultimately are then trafficked to the immune system that stimulate a cascade that leads to specific B cells that are more prone to forming galactose-deficient IgA. It seems that specific cytokines, BAFF and APRIL, play a significant role in those processes that leads to the production of B cells, both mature and plasma cells, that produce the galactose of IgA that leads to the disease state of IgA nephropathy. So, John, can you tell us a little bit more about these key mediators of B cell growth like APRIL and BAFF?
Sure, Sunil. So, how are B cells controlled? What drives B cell proliferation? What drives that class switching to IgA production? And what promotes B cell and plasma cell survival? Well, what's become apparent is that there are two key cytokines called BAFF and APRIL that are essential for B cell homeostasis. And they act on a number of different cell surface receptors that recognize either BAFF or APRIL or both. And that is how these cytokines control the development of B cells and the production of antibodies. And as you can see here, B cells at different stages of development differentially express these receptors. And if we want to control the activity of the whole B cell lineage, then we need to be able to target both BAFF and APRIL because these cytokines act at different stages of B cell development. And that is why, when we're thinking about IgA nephropathy, we're thinking about how we can target both of these cytokines to really reduce the development of these antibody secreting cells that are generating this Gd-IgA1, but also those antibodies that are in the circulation that recognize Gd-IgA1 and amplify immune complex formation. Interestingly, we've performed a number of genome-wide association studies, and consistently in those genome-wide association studies, we found a number of genetic risk alleles that are associated with the B-cell activation pathway. And in particular, two genes that stand out are genetic risk alleles associated with the genes for APRIL and for the cell surface receptor TACI, which binds both BAFF and APRIL, really highlighting the importance of the APRIL and BAFF pathway in driving the production of pathogenic IgA and making it an obvious therapeutic target. What we've also seen is in a number of observational studies that high levels of APRIL are associated with an increased risk of kidney failure. And you can see here on these graphs where we separate out APRIL levels by tertile. Those patients with the highest tertiles have the highest risk of kidney failure and the highest risk of doubling of serum creatinine. Equally, when we look at levels of BAFF in the circulation, higher levels of BAFF are associated with higher degrees of proteinuria, again suggesting that these two key cytokines are critical in driving the glomerular injury and the progressive loss of kidney function that we see in IgA nephropathy. And so we now have a number of agents that can target either BAFF and APRIL or APRIL alone. We have two monoclonal antibodies, sibeprenlimab and zigakibart, that target APRIL. And we have three drugs, atacicept, povetacicept, and telitacicept, that block both BAFF and APRIL signaling. All five agents are currently in phase 3 studies. And so what I'm going to show you here is some very new data on the phase 3 Visionary trial, which has looked at sibeprenlimab in IgA nephropathy. This data was presented at the European Renal Association meeting in early June. And what we see here in this large phase 3 Visionary study, which recruited globally and recruited high-risk patients with persistent proteinuria despite optimized supportive care, which included RAS inhibition and SGLT2 inhibition, was that addition of sibeprenlimab led to a significant reduction in proteinuria. And importantly, when we look at the safety data available from this interim analysis, we can see that sibeprenlimab was very well tolerated and had an exceptional safety profile. We don't know any more data at this present time because this drug is currently, and the data are currently going through the approval process with the FDA. But hopefully in the next 12 months or so, we will see the GFR data as well. Also at the ERA was new data on zigakibart. Now, this is at an earlier stage of development. The data that was presented was 100-week data from the phase 1 study that looked at an open label study of two different cohorts of patients who received zigakibart. The top level data from that study was that treatment with zigakibart led to a clinically significant reduction in proteinuria, which was sustained over the full 100 weeks, suppression of Gd-IgA1, that pathogenic form of IgA, alongside stability of GFR over the full 100-week period. Again, this was a study that recruited patients with IgA nephropathy at high risk of progression due to high levels of residual proteinuria, despite adequate blood pressure control and renin-angiotensin system blockade. So, Sunil, can you take us through now the data for the BAFF/APRIL inhibitors that are in development?
Absolutely. Thanks so much. So the first one we'll talk about is atacicept, which is a small molecule fusion protein that inhibits the binding to BAFF and APRIL. What we have so far is the phase 2b study findings, as well as some look into the future with the phase 3 Origin trial. So what's notable is that both at 24 weeks and 36 weeks, we saw a marked reduction in proteinuria compared to placebo. And then when we looked at the 96-week open label extension data, there was a sustained reduction in galactose deficient IgA. There was further reduction in UPCR. There was preserved renal function, and there was a reduction in the percentage of participants with ongoing microscopic hematuria. The phase 3 trial is currently ongoing, is completed enrollment, and we expect to see the primary endpoint, the reduction in proteinuria, very soon in the second quarter of 2025. And based on that data, we should expect to see a submission for accelerated approval for the second half of 2025. The next agent I'll outline is povetacicept, which is also a small molecule fusion protein that inhibits the binding to BAFF and APRIL. And similarly, we've seen in the phase 2 trial, the assessments at week 48, a reduction of proteinuria by 66%, fairly preserved eGFR with an absolute change of 1.1% only, a clinical remission in 63% of patients, and reduced galactose deficient IgA by approximately 68.9%. Once again, this phase 3 trial is ongoing, the Rainier trial, and we expect that to complete in 2026. Lastly, we see a study only going on in China of telitacicept, which is a small molecule fusion protein that inhibits the binding to BAFF and APRIL. The study design was quite similar to the previous studies, and the outcomes are also similar, where we've seen a 25% reduction from baseline in the cohort receiving the 160 milligram dose, a 48% reduction from baseline in the cohort receiving the 240 milligram dose, and overall, the agent has been well tolerated. Similar to the other trials that we outlined, the phase 3 trial is completed enrollment, and we should expect completion in late 2026.
Thanks, Sunil. So there are many emerging treatments to target the B-cell pathway in IgA nephropathy. We've discussed all of those targeting BAFF and APRIL that are currently in phase 3. Very exciting data. And B-cell therapies target the key components of the pathophysiology of IgA nephropathy, namely APRIL and BAFF. Agents targeting APRIL and BAFF have demonstrated sustained effects on both UPCR and GFR, as well as profound reductions in the pathogenic form of IgA, Gd-IgA1, which we think is the main driver for immune complex formation in this disease.
Thanks so much, John. We seem to be at the cusp of a true revolution in our approach to IgA nephropathy. It's a very exciting time to be a nephrologist and exciting time to be involved in this field. Thanks so much.
So thank you for this great discussion, and thank you for participating in this activity. Please continue on to answer the questions that follow and complete the evaluation.